For basically all of human history, one of the biggest mysteries out there was, what is the stuff of life? What is the actual physical molecule that carries the blueprint for a living thing and passes it down through the generations? Well, today we are diving deep into a scientific detective story to uncover that very secret.
That sounds like a total paradox, right? But it actually captures our story perfectly. For thousands and thousands of years, humans have been practicing genetics. We've been selectively breeding plants and animals, but for almost all of that time, we had absolutely no idea how it worked.
So people knew there had to be some kind of blueprint, right? A set of instructions. But finding the actual molecule it was written on, that was the puzzle. So today, we're going to follow the clues, piece by piece, and crack the case of the most important molecule on Earth.
And to really get a feel for this mystery, you've got to understand just how far back it goes. Our relationship with heredity, it's not a new thing. It goes way back to the dawn of civilization, as you're about to see.
Think about this. Around 10,000 BCE, our ancestors were already deep into genetics. They just didn't call it that. They were breeding better crops, stronger animals. But it took almost 12,000 years for Gregor Mendel to even figure out the basic rules of how this all works. And then nearly another 100 years after that to finally identify the molecule responsible. For millennia, we were masters of the effect, but we were completely blind to the cause.
All right, so the case goes cold for centuries. Then we get our first real detective on the scene, an Augustinian friar who happened to have a real passion for pea plants.
This guy, Gregor Mendel, he was just brilliant. By meticulously breeding thousands of pea plants, he figured out that traits aren't just a random blend. They're passed down in predictable, mathematical patterns. He basically laid out the fundamental principles of heredity. It's like he figured out the entire pattern of a crime, the motive, the method, the timing, but he never identified the culprit. The physical gene was still a total black box.
Okay, so let's fast forward to the early 20th century. Scientists have now zeroed in on the scene of the crime, the cell's chromosomes. And inside, they found two main molecular suspects.
And this is where things get really interesting, because the scientific community basically fell for a classic red herring. On one hand, you have protein. It's complex, it's versatile, it's built from 20 different amino acid building blocks. It seemed like the obvious candidate, right? It had to be complex enough to write the epic story of life. And then there's DNA. It seemed, kind of boring, a simple repetitive molecule made of just 4 little building blocks. The consensus was clear. Protein was the prime suspect, and DNA was just the boring structural stuff holding it all together.
But, just like in any great mystery, the evidence started pointing in a totally unexpected direction. A series of landmark experiments began to build a case that was pretty much impossible to ignore. The first big clue, the first real crack in the case, comes from a guy named Fred Griffith in 1928. He was working with bacteria that caused pneumonia, and he found something incredible. If you took deadly bacteria, killed them with heat, and mixed their guts with live, harmless bacteria, something from the dead bacteria could permanently transform the harmless ones into killers. A physical substance was carrying genetic instructions. The question was, what was it?
16 years later, a team of researchers, Avery, McLeod, and McCarty, finally cornered this transforming principle, and they basically interrogated it. They used enzymes to systematically destroy each suspect. So they destroy the proteins, but the transformation still worked. Okay, they destroy RNA. Transformation still worked. But then they used an enzyme that only chews up DNA. And what happened? The transformation stopped, dead in its tracks. This was the smoking gun.
Now you'd think that would be enough, right? Case closed. But a lot of scientists were still skeptical. The world needed one final, undeniable piece of evidence. And that proof? It came from an incredibly clever experiment involving a virus, some bacteria, and, believe it or not, a kitchen blender.
Alfred Hershey and Martha Chase designed a brilliantly simple experiment. See, the virus they used is basically just a protein shell with its DNA packed inside. So they made 2 batches. In batch 1, they put a radioactive tag on the protein coat. In batch 2, they put a different radioactive tag on the DNA. Then they let both batches of viruses infect bacteria, used a blender to literally shake the viruses off the outside of the cells, and then they just checked to see which radioactive tag actually made it inside.
And the result? It was crystal clear. All the radioactive protein was left floating outside. But the radioactive DNA? That was inside the bacterial cells, already at work, hijacking them to make new viruses. The case was finally solved. DNA, not protein, was the molecule carrying the instructions. The unlikely candidate was the culprit all along.
So the whodunit part of the mystery was solved. But that just opened up a brand new question, a howdunit. I mean, how could this simple-looking molecule possibly store and copy the entire blueprint for life? The answer was hidden in its structure.
And this brings us to one of the most famous moments in all of science. Using the absolutely critical X-ray images produced by the brilliant scientist Rosalind Franklin, images that hinted at a repeating helical shape, two other scientists, James Watson and Francis Crick, were able to finally piece together the puzzle. They built a model that literally changed the world.
And what they found? Well, it's just beautiful in its simplicity. DNA is a double helix, right? Kind of like a twisted ladder. The sides of the ladder are made of sugar and phosphate, and the rungs are made of those four bases, A, T, C, and G. And here is the secret, the absolute key to everything. The pairing is specific. A always pairs up with T, and C always pairs up with G. It's a simple rule, but it is profound.
And in that one simple structure, suddenly, everything clicked. The sequence of the bases along the ladder, that's the code that stores information. And because A always pairs with T and C with G, the molecule can be unzipped right down the middle, and each half can be used as a perfect template to build a new partner. It was a perfect system for both storing and copying information. Case closed. For real this time.
But, cracking the code of DNA wasn't the end of the story. Not even close. It was the starting pistol for a revolution in science and medicine that we are still living through right now. 3 billion. That's a huge number, right? Well, that is roughly how many DNA base pairs, how many letters, A's, T's, C's, and G's, are in the instruction book for a single human being. And thanks to the Human Genome Project, which was finished back in 2003, we now have the complete reference sequence for our own species.
Knowing this code has just completely changed the game. It's unlocked possibilities that our ancestors couldn't even have dreamed of. We're talking about personalized medicine that's tailored to your specific genetic makeup, the incredible promise of gene therapy to fix diseases at their root cause, and just a fundamentally deeper understanding of evolution that connects every single living thing on this planet.
So we've gone from a complete mystery, from knowing nothing about the blueprint of life to holding its complete sequence in our hands. The first great detective story of genetics is over, but now the mystery is a totally different one. With this incredible power to read and even write the code of life, what are we going to build? What will we choose to change? That's the next chapter, and it's one we are all writing together.